Okay, now we are moving on to chapter 3, which is the cellular level of organization. Again, this should still be reviewed from biology, so we're not going to go into massive detail, but we're going to talk about the parts of the cells, plasma membrane structure and function, the cytoplasm and the nucleus, how proteins are synthesized, cell division for somatic cells, and cellular diversity. And then we're going to touch on aging and cells and homeostatic imbalances. So, cells are the basic living structural and functional unit of the body. So what that means is that it's the smallest part of the body that can perform all of the functions of the body. Cytology is the study of cells. Cells are really diverse in their structure and function, but all of them have three main components. All cells have plasma membranes, cytoplasm, and a nucleus. The plasma membrane is the barrier between the internal and external environments. So the plasma membrane is what surrounds the cell. It is regulatory. It's selectively permeable, meaning that it only lets certain things in and certain things out. The cytoplasm is the area between the nucleus and the plasma membrane. This is where the cytosol is contained, and that is composed of water and dissolved solids, and it kind of bathes the organelles and suspends them. The nucleus is the control center, so the nucleus is like your boss. The nucleus tells the cell what to do, when to do it, and how much to do it. This is where the chromosomes are, and chromosomes are made of DNA, and DNA contains genes, which are the hereditary units. So the nucleus contains our chromosomes, which basically dictate who we are, what proteins we will make, and when we will do everything, including certain aspects of behavior. Here's the plasma membrane of a typical cell. This has everything possible in this cell. So there's the plasma membrane surrounding the cell, the cytoplasm between the nucleus and the plasma membrane, and then the nucleus itself. So the nucleus is rather large, as you can see, and we're going to talk more about the specifics later on. So the plasma membrane is known as a fluid mosaic model. It has some fluidity to it, but yet it stays together pretty well. It has an olive oil consistency in between the lipid bilayer, and it's made up of phospholipids. Those blue things with the little white tails hanging from them, those are the phospholipids. The yellowish little things between the phospholipids, that is cholesterol. And then you have some proteins, which you can see are the large orangish things, and the purpley things are carbohydrate side chains, which we'll talk about those later on. So all of these patchworks are what give it the mosaic pattern that it is known for. So there's the external versus the internal. That is the specific phospholipid bilayer. Cholesterol, as I said, glycolipids, and carbohydrate chains. An integral protein, integral means that it goes all the way through the plasma membrane, so it goes all the way through it. And peripheral proteins are proteins that do not go all the way through it, they just kind of hang out on the one side or the other of the plasma membrane. And then glycoproteins. Cytoskeletal fibers are fibers that kind of hold it in place and also allow other things to attach. And carbohydrate chains, as I said earlier. So, what's the function of all of these things? Again, the cholesterol aids in the fluidity and selective permeability. The cholesterol, when it gets warm, the plasma membrane has a tendency to become really fluid, so the cholesterol molecules will actually kind of grab on to the phospholipids and make sure that they stay relatively close together. And on the other hand, if it gets really cold, the plasma membrane has a tendency to become more solid, so the cholesterol molecules will actually push and spread the phospholipids apart. The glycolipids and glycoproteins function as cell markers. It actually gives the cells identity and allows for histocompatibility testing and self-identification. Basically what that means is that it allows our immune system to recognize self-cells so that we do not attack, our immune system doesn't attack our own cells. And histocompatibility testing just means if you match somebody else to donate an organ or donate blood to, or bone marrow, things like that. Integral protein functions vary. Some of them are channels, meaning they have an entire hole all the way through them so that certain ions can pass directly through. Some of them are carrier proteins, meaning that they transport certain ions or molecules like glucose, for example. Glucose can attach to a carrier protein and then be taken into the cell. Some of them act as receptors, so hormones can bind to them and then be brought into the cell. Some of them function as enzymes, so they will speed up, catalyze reactions. Some of them are linkers, meaning they just kind of hold cells together, and others still function in cell identification. So here's a chart just summing up all of the functions of the proteins. So there are 6 of them. So movement of substances is going to occur between the intracellular fluid and the extracellular fluids, as we said in the last chapter. The intracellular fluid, like the cytosol, and the extracellular fluid, like the interstitial fluid, which is the fluid between the tissues. Substances can move in and out of the cell 3 different ways. They can go directly through the phospholipid bilayer. They can use an integral protein, using either a channel protein or a carrier protein. Or they can go in by vesicle formation, which is known as bulk transport, which we'll talk about later. Things that affect how they're transported. The concentration gradient is a big one. Molecules naturally flow from their high level of concentration to a lower level of concentration. If they have to go from a low area of concentration to a high area of concentration, they're going to need active transport, so that's going to require energy. Size is going to, of course, be a factor. Larger molecules need a lot more help than smaller molecules do. Charge is going to be a factor. Ions need help oftentimes than uncharged particles. And lipid solubility, how soluble something is in a lipid. Polar molecules there. So all of those are going to have to be helped getting across the membrane because of those factors. So we have 2 basic categories of transport. Passive transport. Molecules are moving with or down the concentration gradient until equilibrium is met. It does not require ATP, so it's passive. If you think of when you're active, you're using energy, so active transport requires ATP. Passive then does not. Some examples, simple diffusion. There isn't an integral protein involved. The molecules simply pass right through the plasma membrane, like carbon dioxide and oxygen, for example. Or facilitated diffusion, where an integral protein is required. So it can either be a channel protein or a carrier protein, but they will go right through. Osmosis. Anytime you hear osmosis, think of water movement. Osmosis is the diffusion of water molecules across the permeable membrane. So they will use what are called aquaporins, which are water channels. And the water will do the moving, not the solutes themselves. So the molecules will not move. It's the water that moves. So anytime you hear osmosis, think diffusion of water. Tonicity refers to the relative solute concentration of a solution as compared to another. So we can have 3 different types. Isotonic is when both have equal concentrations of solute. Hypotonic and hypertonic. Hypotonic is when there is a higher concentration of solutes inside the cell than outside the cell. So water will move into the cell to try to dilute the cell down, and the cell will actually swell up. So if you remember hypo, like the O, like the cell's swelling, maybe. Hypertonic is the opposite. Hypertonic, there are more solutes outside the cell than inside the cell. So water will move out of the cell to try to dilute the surrounding area. This causes the cell to shrink or crenate. So if you remember polyper. Now, if you think about it, water is what's doing the moving. So water is still going down its concentration gradient. It's trying to go from an area where it is more concentrated to an area where it is less concentrated. So water and solute concentration are opposite. So if you have more water, you have less solutes. If you have less water, you have more solutes. So water is moving from where there's more water to where there's less water. So here's just a picture showing you simple diffusion. It goes straight through the plasma membrane. Channel-mediated facilitated diffusion, it goes through the channel. Or carrier-mediated, it attaches to the carrier protein and then is brought into the cell that way. So water always flows from a hypotonic environment to a hypertonic environment. The greater the difference in the concentration gradient between the solutions, the faster the osmosis will occur. And that's the case with everything. The greater the difference, the faster it happens. So this is going to increase the osmotic pressure. So the cytosol of erythrocyte contains about 0.9% NaCl and 5% glucose. So if you have in the pictures below intracellular fluid versus extracellular fluid, if you have a hypotonic environment outside the cell and a hypertonic inside the cell, water is going to go inside the cell and the cell is going to swell and the cell will burst. On the other hand, if you have a hypotonic environment inside the cell and hyper outside the cell, water will leave the cell and the cell will shrink or crenate. So when the cell swells, it's called hemolysis and it'll burst. When the cell shrinks, it's called crenation. For red blood cells we're talking about. So isotonic, you have a normal red blood cell shape. Hypotonic, water goes into the cell and the cell will swell, possibly burst. Hypertonic, water leaves the cell and the cell will shrink. Active transport requires energy expenditure, so ATP. Primary active transport is when the molecules are moving from low to high concentration, so going against the concentration gradient. It requires ATP and a carrier protein. The sodium-potassium pump is probably the best example. We have to maintain the sodium and potassium concentrations around our cells in order for action potentials to occur properly. So we normally have concentration of sodium outside the cell and then a low concentration of potassium, inside the cell. So what happens when an action potential occurs is sodium actually rushes into the cell. But since there's a high concentration of sodium out there already, we have to pump it back out eventually. So it has to go from low concentration to high concentration. So how do we do that? Well, active transport. So the sodium-potassium pump will pump 3 sodium out and 2 potassium in to get the original ion concentration so that everything can return back to normal. Secondary active transport is kind of when somebody hitches a ride. So protons, for example, will hitch a ride. Sodium will hitch a ride, but other things will hitch a ride when something else is being transported. So you have to maintain that electrochemical gradient and tonicity both inside and outside the cells in order for a cell to function properly. So here is the sodium gradient and the potassium gradient, and it's just showing you the sodium-potassium pump. So what happens is 3 sodium will be pumped out into the extracellular fluid, and in the meantime 2 potassium will be pumped into the cytosol, basically. So as you can see, if you look at the pictures, ATP comes in and phosphorylates the ATP pump, and then, or the sodium-potassium pump, sorry, and then that phosphate goes away, and the whole process will start all over again. Bulk transport is how large molecules get in or out of the cell, and it's through vesicle formation. Endocytosis is what brings substances into the cell. Exocytosis releases substances out of the cell. So endocytosis actually has 3 different types: receptor-mediated, phagocytosis, and pinocytosis. Receptor-mediated endocytosis is highly selective. Basically, you can transport things like low-density lipids, transferrin, hormones, things like that. But it will actually bind to the receptors and then be transported. Phagocytosis is how you get larger molecules inside the cells, like bacteria. And what happens is the plasma membrane actually kind of puts pseudopods out and reaches out, grabs whatever it is, surrounds it, and brings it in. Pinocytosis is also known as drinking, and this is how importation of smaller solute molecules comes in. What happens is the plasma membrane kind of invaginates on itself and then surrounds whatever we're bringing into the cell and then brings it in. Exocytosis is the reverse of this. So whatever substance we want to get out of the cell, we surround it with a vesicle. It goes to the plasma membrane, fuses with the plasma membrane, and the contents are let out into the extracellular fluid. So the loss of the plasma membrane during endocytosis is recovered through exocytosis. So the part of the plasma membrane that surrounds the molecules coming into the cell is replaced when you put things out of the cell. So here's a picture showing you receptor-mediated endocytosis, bringing in an LDL. You can see it uncoats, it fuses with a lysosome, and it's degraded. And then we can recycle some portions. Phagocytosis, as I said, the pseudopods kind of come out and grab whatever it is, surround it, and bring it in. And pinocytosis, the plasma membrane invaginates on itself and then surrounds whatever is bringing into the cell. So the cytoplasm is the area between the plasma membrane and the nucleus. The cytosol is the fluid portion of the cytoplasm. Organelles are the structures that perform specific functions. Usually they're bound by membranes. And then the nucleus is the regulator. It regulates all the activities and contains our DNA. So the cytoplasm contains cytosol, which bathes the organelles that perform specific tasks and kind of suspends them. And the nucleus is the control center. This is involved in mitosis and other activities like transcription and translation. So the nucleus, as I said, is like your boss. It tells our bodies and our cells exactly what to do at all times. So make sure you can identify various organelles and their structure and function for your test. Oops, sorry. An overview of protein synthesis. So if we want this particular gene segment to be expressed, what's going to happen is the DNA will transcribe a piece of messenger RNA in the nucleus, and then that messenger RNA will leave the nucleus and go to the cytoplasm, where it will bind to a ribosome to be translated into a protein. Some terms you need to know for your test: transcription, the process that makes RNA from a segment of DNA. RNA polymerase is the enzyme that catalyzes transcription. The promoter region is the place on the DNA where RNA polymerase binds to start transcription. The terminator is where transcription ends. And translation is the process that builds the polypeptide from the RNA, so that's making the protein. DNA is the molecule that contains the units of heredity, which are our genes. Messenger RNA is the molecule that contains the codons. Transfer RNA is the molecule that contains the anticodons and brings a specific amino acid to create the polypeptide. And ribosomal RNA matches the codon to the anticodon, and that's what ribosomes are made of. So again, this should all be reviewed from biology. That's why we're not spending a whole lot of time on it. The codon is the triplet code, which is 3 nucleotide base sequences on the messenger RNA, and each codon represents a particular amino acid. And then the protein will be built by translating those codons and attaching the amino acids together. So here you see step 1 is transcription. The DNA creates a piece of messenger RNA. That messenger RNA leaves through the nuclear pores, goes into the cytoplasm, where it binds to the ribosome at the bottom there and creates the protein by translation. Here is a closer up view. And here it all is translation and bigger picture. So the central dogma just refers to the fact that DNA is transcribed into RNA, which is translated into proteins. Multiple transcripts of messenger RNA can be made as long as the DNA is unzipped. Messenger RNA transcripts are edited in the nucleus before translation occurs in the cytoplasm. So what happens is the introns are cut out and the exons stay in and are spliced together. So the exons are what are actually expressed. And you can get multiple transcripts by splicing it in different ways. And again, review from biology. Messenger RNA transcripts may be translated multiple times and have more than one ribosome attached. It's called a polyribosome. And the synthesis of proteins is fast and accurate. Now, if you do not remember those main steps, you should probably review some biology. Now we're going to talk about cell division, which again is review from biology. Somatic cell division is called mitosis, and it produces genetically identical cells from generation to generation. So your parent cell, your original cell, is diploid, 2N, 46 chromosomes. Your daughter cells will be diploid, 2N, with 46 chromosomes, and they are identical to the parents. So the purpose is growth, repair, and specialization, or differentiation. So some terms: somatic cells are all the cells in the body except for the germ cells, which, remember, the germ cells are your sex cells, so the ova and the sperm. Diploid denotes a full set of chromosomes, so 2N. Mitosis is the division of the nucleus, and cytokinesis is the division of the cytoplasm. Meiosis occurs in the germ cells, so your ova and your sperm, and those are found in the gonads. And this is reproductive cell division that results in the formation of gametes. So mitosis is cell division in the somatic cells, where 2N, the diploid, goes to 2N, the diploid. Meiosis occurs in your germ cells, where your 2N goes down to N. So the diploid goes to haploid. The point of meiosis is to reduce the number of chromosomes in half, so that when the egg and the sperm fertilize, you have a complete zygote. Some terms associated with DNA. Chromatin is very thin, and that is what you can see. Kind of looks like spaghetti in the nucleus. Chromosomes are the condensed chromatin. It actually condenses into chromosomes. What happens is it wraps itself around the histone proteins. Chromatids are the chromosomes in their replicated state. They have 2 chromatids, each one, like so. They're joined at the centromere. The cell cycle is the sequence of events that occurs when the cells are duplicating. So as you can see over at the side here, you have the G1 phase, the S phase, the G2 phase, and the M phase. The G1 phase is the phase where they're getting ready to divide. The cells are active. They are duplicating their organelles, duplicating the cytosol components, making sure that they have everything they need for cell division to occur. The S phase is the DNA replication phase, so the synthesis phase. The G2 phase is when the cell's continuing to grow, but now it's double checking and making sure that it has everything it needs to successfully complete mitosis. And all those 3 together, the G phase, G1, S phase, and G2 phase, are called interphase. Our cells spend most of the time in interphase. The last phase, the mitotic phase, the M phase, that's when the nucleus is actively dividing. And that's divided into 4 sections: prophase, metaphase, anaphase, telophase, or PMAT. So some cells are continuously entering into the cell cycle and divide a lot, like epithelial cells, stem cells, and unfortunately cancer cells. Some cells enter into the cycle only when needed, so like liver cells if we need to repair our liver. And some cells, once they leave, never return, like nerve cells and skeletal muscle cells. Once your nerve cells are damaged, they pretty much stay damaged. They can't repair themselves. So again, interphase, the S phase follows the semi-conservative model of replication. The DNA unwinds. Helicase is the enzyme that breaks it apart. Complementary base pairing occurs. Adenine to thymine, guanine to cytosine. The new and old strands are joined together, and then we have a proofreading stage. So again, if you don't remember replication from biology, you might want to review it real quick. Prophase is the first phase of mitosis. The chromatin condenses into chromosomes. The nucleolus disappears. The nuclear envelope starts to fragment. The centrosome moves to opposite poles, and spindle fibers begin to form. So in this phase, you can actually see the chromosomes inside the nucleus. In metaphase, the replicated chromosomes align at the metaphase plate, which is the middle. So if you remember metaphase middle, you should be good to go. And the spindle fiber formation is complete. In anaphase, the centromere separates. The single chromosomes migrate to opposite poles, and cytokinesis actually begins by the cleavage furrow. So in this phase, you can see they look like little V's being pulled to opposite poles. And in telophase, it's basically the opposite of prophase. The chromosomes go back to chromatin, the nucleolus reappears, the nuclear envelope rebuilds, spindle fibers disappear, and cytokinesis is also almost complete. You can see that cleavage furrow really developing. Cell signaling occurs by chemicals, so that's what's going to determine if the cells live but don't divide, like if they stay in the G0 phase, grow and divide, or die and undergo apoptosis, which apoptosis is programmed cell death, so they basically commit suicide. Tumor suppressor genes and proto-oncogenes control the cell cycle. And cells that can't control their cell cycle can result in tumor growth. Cancer cells grow uncontrollably and uninhibited. So if cells can't control their cell cycles and do that, they can result in cancers. Tumor suppressor genes, just so you know, are exactly what the name implies. They suppress tumor formation. So if you have a mutation in a tumor suppressor gene, like the P54 gene, for example, that can really lead to cancer. half of all cancers actually have a mutation in the p54 tumor suppressor gene. Don't worry about meiosis. So you're responsible for cellular diversity on page 98 in your book, aging and cells on page 98 in your book, and then homeostatic imbalances on 99 to 100, and medical terminology on page 101. I just want to say a couple of things about cellular diversity. All it really refers to is the fact that cells are diverse. You have neurons, you have muscle cells, you have different types of epithelial cells, you have blood cells, you have connective tissue cells, which are a variety of those. So cells can be very diverse, and they can look very diverse and have diverse functions. For example, let's take a squamous epithelial cell. Let's say it's stratified. So it's going to function in protection, it's going to be layered, and it's going to be in areas subject to abrasion, like your skin, for example. And then let's take a neuron, a nerve cell. They are shaped completely different. They have dendrites, a cell body, an axon, and they generate impulses and carry those impulses where they need to go. Then let's say a muscle cell. Muscle cells, again, completely different shape, and they function in contraction. So cells are highly diverse in both structure and function. And remember, structure and function go hand in hand. So they're shaped for what they need to do. As far as aging goes, there's a few theories on aging. One of those theories is called the wear and tear theory, which basically just states that we get old. Our bodies age because we use them. You know, it's a fact of life. We get older, we use our bodies, some of us more than others. Damage is going to happen. Other theories say that cells only have a limited number of times they can divide. Once they hit that number, if there's no replacements, well, we age and we die. Another one says that on the end of our chromosomes are something called telomeres. And every time the cells divide, those telomeres get shorter and shorter. So once the cell divides so many times, those telomeres, those ends of those chromosomes are getting shorter and shorter. So now you're actually starting to lose genes and things that are important for survival. As far as homeostatic imbalances go, the biggest thing is cancer. Cancer cells grow uncontrollably. They do not exhibit contact inhibition, so they grow right on top of each other. They lack specialization and differentiation, so they just kind of, fit in anywhere. And they experience something called angiogenesis, which means that they grow their own blood vessels to these tumors that are formed. If the tumors become metastatic, that means they branch off from where they originally began and travel to other areas of the body. It can be very deadly. If angiogenesis occurs and blood supply is getting fed to these tumors, that is very deadly. And that's one reason why, if you've ever watched any of those shows where people have tumors all over and the doctors are saying it's difficult to remove them, almost impossible sometimes, it's because of the blood supply that goes to these tumors. If you just cut it off, you're going to have the patient bleeding out because you have to stop that blood supply somehow. So they're trying to develop medicines that actually prevent angiogenesis from happening. And that would probably be a really great thing. Some of the tumor or cancers we're going to talk about throughout the semester and then next semester if you're taking that. And a lot of the tumors that are cancers that people have are cancers of the epithelial tissue. Melanomas are probably one of the worst skin cancers that you could have. And we're going to talk more about that in Chapter 5. carcinogen is basically anything that can cause cancer. So oftentimes you hear about all the carcinogens in cigarette smoke and cigarettes in general and, various things. But the thing is, people have had lung cancer that have never smoked a day in their lives. So homeostatic imbalances are one of those things that sometimes it's hard to predict who they're going to happen to and why they're going to happen, especially when it comes to things like cancer. Okay, and then you're supposed to go through the medical terminology as well. So like atrophy, hypertrophy, when cells shrink in size or increase in size, things like that. So make sure you look that over. Okay, so that is all. If you have any questions, send me a message. Bye.